Thank you. Great. Thank you. So I'm Sally Morton. I'm Dean of the College of Science, and I have the honor of introducing our speaker today. The host of today's talk is the Department of Biological Sciences Diversity Committee, who have been leaders in this series for many years, especially during the university's celebration of Dr. Martin Luther King Jr. The list of co-sponsors is a long one, but I think the length of the list indicates the support for today's speaker, the seminar series, as well as the university's celebration this week of Martin Luther King. So let me read the co-sponsors. The College of Natural Resources and the Environment, the College of Veterinary Medicine, and the College of Science. In particular, our college's diversity committee, as well as our Department of Biological Sciences, the Virginia Tech Life Sciences Seminar, the Ecology, Evolution, and Behavior Seminar, the Women and Minority Artists and Scholars Lecture Series, the Global Change Center, and the Virginia Tech Office of Inclusion and Diversity. Let's thank our co-sponsors. So a core key value of the Virginia Tech College of Science is inclusion and diversity of not only people, but ideas. A welcoming, inclusive, and respectful environment is not only beneficial for good science, I think it's essential. Diversity makes science, our university, and our society strong. At Virginia Tech, as stated by Dr. Menya Pratt-Clark, who's the Vice President for Inclusion and Diversity, we are committed to change, to sustainable transformation, and to actualizing Virginia Tech's vision of providing access and education to students, all students, with the potential and ability to transform the world. And we're very grateful to Dr. Vanessa Ezenwa, our visitor today, our speaker today, your willingness to be part of our celebration of Dr. Martin Luther King. Dr. Ezenwa is a distinguished scientist who is a professor in the Odum School of Ecology at the University of Georgia, with a joint appointment in infectious diseases in the university's College of Veterinary Medicine. She received her BA in biology from Rice and her PhD in ecology and evolutionary biology from Princeton. She was a postdoc at the United States Geological Survey, USGS, and she began her academic career at the University of Montana, and she moved to Georgia in 2010. She studies the ecology of infectious diseases with a specific focus on linking processes across scales. And I spent some time Googling her. And I found a quote that I like. Here's a quote. "I find parasites fascinating." How can you not love that? So what interests me most, quoting Dr. Ezenwa, what interests me most about my field of research is learning about theories and approaches from different disciplines and combining these to advance our understanding of how cryptic parasites influence everything from behavior, physiology, and ecology, and evolution of their hosts. So I think we're going to hear more about the combining of approaches today. I'm looking forward to that. Among her numerous accolades, Dr. Ezenwa received the Curro Research Mentoring Award in 2019, the Creative Research Award in 2016, and she was a Fulbright Scholar in 2014. She is dedicated to her students, and I found in another interview at Georgia, she spoke about one of the highlights of her career is when she takes students to Africa to conduct field research. Now, as I prepared for today, I personally reflected on the unfinished vision of Dr. Martin Luther King, Virginia Tech's opportunity to contribute to this legacy, and our own individual responsibility. Dr. King wrote a student paper at Morehouse College on the purpose of education. He wrote it in 1947. He was 18 years old at the time, and I quote, "Education must enable one to sift and weigh evidence, to discern the true from the false, the real from the unreal, and the facts from the fiction." He went on to say, "We must remember that intelligence is not enough. Intelligence plus character, that is the goal of true education." So thank you all for contributing to the educational mission of Virginia Tech and for helping all of our students achieve the goal of a true education. And thank you, Dr. Ezenwa, for joining us today. I know that we're in for a rare treat as Dr. Ezenwa presents her talk titled Worms, Germs, and Buffalo, a co-infection story. Please join me in welcoming Dr. Ezenwa.
Thank you. Okay, so first I want to thank Sally for that very gracious introduction. And thanks to the Diversity Committee and all of the co-sponsors that were mentioned for inviting me to this presentation, which is a real honor. And so today I thought that I would tell you guys a story about some of the a main theme of research in my lab, and sort of end with a quick sort of summary of how I came to be doing this kind of work. And so parasites are fascinating. I'll start with that. And they're everywhere. And one thing that I sort of learned early on when I started working with parasites as a graduate student is that the co-occurrence of parasite infections of different species within the same organism is the norm rather than the exception, right? So most animals, humans, hosts out there are simultaneously infected with a range of different parasite species at the same time. And this graph just highlights a really nice example of this. And these are helminths or worm parasites and protozoan parasites, so things like malaria parasites, infecting humans who live in a single village in the Côte d'Ivoire. And the x-axis here shows the number of different parasite species infecting different proportions of humans in this village of different age classes. So from under 5 to people older than 40. And what we see immediately here is that most individuals that are older are actually infected with far more than one parasite. So even early on in life, so by the age of 5 to 9 years old, this second bar, the majority of these children are already infected with more than one parasite. And this pattern just gets more extreme as you age. So for those individuals that are 40 or older, right, the majority of them, or over 90% of them, are actually infected with more than 4 or 5 parasites. And this is just a small fraction of the parasite diversity that can exist in the human population, right? Just worms and protozoa. This is not accounting for viruses or pathogenic bacteria. So what this really says is that we're sort of a wandering zoo of parasites out there. And just like a plant community or any animal community, so free living communities of organisms, we might expect these parasites to interact when they're inside of a host. And so one thing I'm really interested in is how do these interactions work? Which parasites interact with one another when they co-occur? And what are the consequences of those interactions for both the host and the component parasites? And so a parasite group I'm particularly interested in are the worms. And this is because worms or helminths are extremely common. And so worms infect a majority of vertebrate and invertebrate hosts out there. And using humans as an example, a third of the world's human population are actually actively infected with these worm parasites. So things like this parasite, which is Ascaris lumbricoides, the typical roundworm in human populations. So this graph here is actually a graph of human infections with different types of worm parasites. So as you can see, millions of people out there are infected with these worms. Likewise, you know, there are whole books about the worm parasites of wildlife. And we also know that worms are a huge infection risk for domestic animals, for instance, and cause innumerable production losses in things like sheep and goats. And so another really interesting thing about these worms is not only are they really common, and their being common means that they have a high probability of co-occurring with a lot of other parasites and pathogens, right? So the potential is there for them to co-occur or co-infect simultaneously as a lot of microbes. So how do they influence these other parasites with which they co-occur? So one reason worms are particularly important when we think about co-infection is that there's reasons why we should expect that worms might actually have a profound effect on the other types of parasites and pathogens for which they co-occur. So emerging evidence for several years now has told us that worms can have a profound effect on the immune system of their vertebrate hosts. So this is the cover article in Science a few years ago about how worms might weaken immunity and impact viral infections in a mouse model. And so an example of how this worm suppression works is that we can think about the vertebrate immune system in a simplified way as sort of having two main arms. One arm that fights off intracellular parasites, so things like viruses and some bacteria. And then another arm that fights off these large extracellular things like the worms. And so unfortunately, these two arms are mutually inhibitory, such that mounting a strong immune response to a worm infection might lead to sort of the upregulation or mounting of a strong immune response to protect yourself against those worms. So things in this part of the scale. But unfortunately, they tip the balance away from those immune responses that are protective against certain viruses and bacteria. So in the public health world, right, a lot of people have thought about the potential consequences of this immune suppression caused by worms. So essentially, by tipping the scales away from immunity that might protect you from certain microbes, it might lead to the exacerbation of key microbial infections, so things like HIV in people, or tuberculosis, right? It might increase your susceptibility to getting infected with these pathogens in the first place. It might lead to faster disease progression and more severe symptoms. And so this immunological paradigm that's been studied quite well in biomedical models and to somewhat in humans has led to sort of ideas about the implications of this worm-based immunosuppression for disease management and control. And so researchers in the field have posited, right, that potentially if we could eradicate these pervasive helminth infections in humans, we might be able to change the face of morbidity and mortality due to parasites like HIV or tuberculosis. So the sort of the big diseases of people, right? So that leads to this question, right? So to control helminth HIV co-infection, should we deworm? And by deworming, can we, on the one hand, protect people from the direct negative effects of these worms, because we know the worms themselves have effects like causing malnutrition and stunted growth in children. But then we might also gain this indirect benefit that by removing these pervasive worm infections, we actually reduce morbidity and mortality rates due to some of the most sort of important human diseases. And so this sort of is out there as a potential hypothesis, like, can we have this win-win? We have effective drugs and fairly cheap drugs, inexpensive drugs for controlling worm infections. Microbes like HIV and TB are particularly difficult to control. So perhaps we could use this indirect strategy of controlling worms to control these sort of challenging microbial infections. So that's a great idea. And we have the drugs potentially to do this. And this is being done sort of at some scale out there because, as I said, the worms themselves cause negative impacts, right? And there's freely, available, effective, and fairly cheap drugs. So companies like GlaxoSmithKline, the pharmaceutical company, actually donate 1 billion tablets per year for mass drug administration programs in parts of the world, right? And these administration programs are coupled with educational campaigns by organizations like the World Health Organization and the USAID to sort of roll out these drugs in communities to try to combat the negative consequences of worms. And potentially, it's this win-win where we reduce the negative consequences of worms caused directly on the host, but we indirectly improve outcomes of HIV and tuberculosis. And so this is sort of the human part of the story, but if we think about domesticated animals, we have the same problem. Worm infections cause massive production losses in sheep, goat populations around the world, and drugs have been a massive sort of tool that we use to control worms worldwide. But the massive application of anthelmintic or deworming drugs to controlling worm infections has led to massive drug resistance problems for these anthelmintic drugs. So alternative strategies for controlling worm infections in domesticated animals have become more and more important. And one of those strategies is breeding these animals for resistance, because resistance traits are fairly heritable, right? So resistant animals that are naturally resistant to these worm infections can pass this on to their offspring. And potentially, like in this graph here, we can breed domestic sheep populations that are fairly resistant and require less use of, right, pharmaceuticals interventions. So these are the main ways that we can control worm infections: drugs and breeding in human and animal populations. But one question that arises, given those interactions that I talked about that happen via the immune system, is what are the consequences of this worm control for microbial pathogens, right? So if these worms have such profound consequences for the immune response for the host, potentially controlling worms is this huge positive benefit where we get rid of the negative effects of worms and we enhance sort of production and reduce morbidity and mortality, potentially for a whole range of microbial pathogens, if it works as we expect. And so given that, I've been interested in this question of whether, right, what sort of the immunological mechanisms tell us, if that really plays out, can we reduce the burden of microbial infections simply by controlling worms? So this has been sort of a guiding question of some of the research we've been doing in my lab, is understanding how these potential interactions between worms and certain microbes might play out in terms of sort of the dynamics of microbial pathogens. So we've been doing this work by studying worms and bovine tuberculosis in wild African buffalo. And bovine tuberculosis, which is caused by Mycobacterium bovis, is a close relative of human TB, which some of you in this room may have had. And so it's bovine TB is what is a zoonotic disease, which essentially means that humans can get bovine TB as well. So we can catch TB from cattle, bovine TB, and cattle can actually contract human TB from us, right? So it's a pathogen that's highly mobile between species, and almost all warm-blooded mammals can acquire mycobacteria in this complex. And so interactions between worms and tuberculosis has been a topic of study in sort of the biomedical literature for some time. And lab rodent studies and human studies suggest that there is indeed some interaction that's immune-mediated that occurs between these two pathogens. What we don't know is what are the larger consequences potentially of these immunological interactions that have been described. We also don't know if those interactions actually play out in a natural context, sort of in the wild or in free-ranging organisms, which is what humans are. And so these wild buffalo are a nice sort of wild model of this for several reasons, right? They're closely related to cattle, and cattle are the hosts of bovine tuberculosis that's very well studied. And so studies on domesticated cattle provide a lot of background information and resources for us to understand bovine TB in these wild relatives of cattle. The other reason this system is quite interesting is that bovine tuberculosis was introduced into our study region, which is where we study this, is in South Africa. So bovine TB was introduced into sub-Saharan Africa by cattle. It jumped into buffalo, and buffalo are now the wild reservoirs who maintain it on the landscape and transmit it back to cattle as well as to people and other wildlife species. So there's a huge concern about controlling and managing tuberculosis in buffalo among managers. Buffalo also, we know, have multiple gastrointestinal worm species that they host. Because they're closely related to cattle, many of their species are quite similar and highly related to the worms of domestic cattle. So again, we have resources and background information that allows us to sort of have more information than we otherwise would in a wild system about the sort of primary actors in this system. So what I'm going to talk about for the rest of this talk here is work we've been doing in Kruger National Park, where TB invaded in around the late 1950s, early 1960s, on how two main control strategies we might apply, so Anthelmintic treatment, so deworming animals, and host resistance, which relates to whether we might breed animals for resistance, how these two control strategies impacts the dynamics of bovine tuberculosis. So with respect to the Anthelmintic treatment study, so essentially our goal with this study was to ask one sort of overarching question. What's the impact of treating or deworming an animal on the outcome, the individual outcomes of TB? So whether they get infected with TB in the first place, and if they are infected, how severe that TB becomes, and what's the effect at the population scale in terms of transmission of TB between and among individuals. So to do this, we set up a study where we captured 200 free-ranging buffalo, and they received radio collars so we could follow them around, and we tracked them for 4 years, essentially to estimate the impact of treatment on these parameters. So the risk, how treatment affected the risk of infection, and how treatment affected disease-induced mortality, so your likelihood of dying of the TB infection. We tracked 2 buffalo herds in the southern part of Kruger National Park over this time period.
Sorry, Vanessa, one quick question. Is this one intervention, or you do this intervention with the drug multiple times?
Oh, yes, so this is the next slide. Good question. And so this is how the study design looks. So essentially, we had 100 treated animals and 100 controls. They were all TB-free at the beginning of the study, right? So they were not infected with TB. They were also all female for logistical reasons. And each animal was caught twice a year, so approximately every 180 days. And at each capture, the treated animals received their drug. So this drug is in a long-acting formulation, which is called a bolus of a drug with the ingredient being fenbendazole that is an anthelmintic drug. This bolus is about this big, if you can see this, and it's a cylinder. Because these are ruminants, that cylinder sits in the rumen, and it slowly releases drug over about a 6-month period. So over that period, it releases drug that helps clear the worm infections, and animals were retreated every 6 months throughout the entire study. So the controls received no treatment, and at the time of each capture, so twice a year, we sort of focused on 3 main variables that we were interested in measuring in each animal. So first, we were interested in whether the treatment was effective, and we measure that by quantifying the number of worm eggs that are being shed in the feces of these hosts because these worms live in the gastrointestinal tract. When they reproduce, their eggs come out through the host feces, and that's how the cycle continues. So we could screen whether our drug was effective by monitoring shedding of worm eggs. The other thing we were really interested in was the immune response to tuberculosis. So essentially, immunological theory tells us that worms are suppressing the immune response to things like TB. So we wanted to measure whether there was a quantifiable effect of treatment on the immune response that they would mount toward TB infection. And third, we measured their TB status, right? So we wanted to know who acquired TB throughout the course of the study. So to answer our first question, we started by being interested in the immune response. So what impact does treatment have on your anti-tuberculosis immunity, your ability to defend yourself against TB if you're challenged with it? And so to test this, essentially we took blood samples from treated and untreated individuals, and we challenged those blood samples in vitro with a mitogen that sort of mimics a TB challenge. And we asked who mounted a stronger response. And what we found was that these bolus animals who received that treatment mounted a stronger anti-tuberculosis response. So essentially, they mounted a response measured by this immune messenger molecule that we know is important in the control of TB infection from mouse and cattle studies. And so the answer to our first question is that much like those laboratory studies that inspired us, we actually see in this free-living wild animal that worms are indeed suppressing the immune response that's important for control of certain microbial pathogens. So the next question was, so if the worms can do this, what impact do they have on the outcomes of infection? So in this free-ranging population, we essentially asked, if manipulating your worm burden can change your immune response, does it have any impact on your likelihood of getting infected with tuberculosis? And so, to our surprise, despite this immunological effect, we found no impact on susceptibility to TB infection. So whether you were treated or not treated, there was no impact on your probability of being infected. And so that's illustrated by this graph here, where on the x-axis, it's time in days since the beginning of the study, and then the proportion of individuals who acquired TB over the course of the study period. Controls are in blue and treated animals are in red. And as you can see from these interdigitated lines, so essentially there was an equal probability of getting TB whether you were treated or not. And so despite the improvement in your anti-tuberculosis immune response, if you were treated, that didn't translate into protection from tuberculosis. So our next question was, well, what happens when you do get TB? So there's no difference in your likelihood of getting it, but perhaps being treated for worms actually allows you to control the TB infection better. Indeed, we saw a drastic effect in terms of the severity of your TB infection, depending if you were treated or not. And in this case, treated individuals were 9 times more likely to survive their tuberculosis infection. So that's a massive benefit of having the Anthelmintic treatment. So just to illustrate that point here, the x-axis is time in days since you acquired your TB infection till you died or the end of the study. And this is the proportion of individuals who survived after acquiring TB. Controls are in blue again and treateds are in red. And as we can see here, this huge separation in these lines sort of illustrates the point that if you're treated for your worm infections and you get TB, you're better able to control that infection and survive your infection. And so this really reveals the massive benefit of deworming for this microbial infection, right? So at an individual level, getting rid of your worms is really good in terms of surviving your tuberculosis infection. But as ecologists, we were also interested in what these individual level effects mean at the population level. So when we think about the control of infectious diseases, we can think about that at two scales, right? We want to control disease at the individual level to protect sort of the individual, right, to reduce symptoms and severity of individual infections. But we also want to control disease at the population scale, right, to prevent the transmission of pathogens between individuals and moderate the prevalence of infections. So we were interested in what these results at the individual level meant for the population level spread of tuberculosis. And so to look at that question, we estimated the basic reproductive number of tuberculosis in our experiment. So basically, we measured this thing, the R0. And you can think about R0 as simply the population growth rate of a parasite, right? And one way we can think about it is that the R0 reflects the number of new infections that a single infected individual will create over the course of their lifetime or over the course of their infection. And so this figure here shows the R0 for some common human diseases, which tells us a little bit about the speed at which different diseases might spread. So we have something like Ebola, which we know can spread really fast. And Ebola has an R0, measles, sorry, and measles has an R0 of 18. So a single infected measles patient can infect 18 others over the course of their infection. And you can compare that to Ebola, which we know doesn't spread as fast, where a single infected individual is infecting only 2 others over the course of their infectious period. And so what we were interested in is what impact treatment has on the R0 of TB. And to do this, we compared the R0 for our subpopulation that were treated for worms and the subpopulation that weren't treated. And what we found essentially when we made these calculations based off of our experimental data was that if you were a control buffalo that was not treated for your worms, so just the average buffalo out there who would get TB infection, your R0 was about 2. So over the course of that individual's infection, it would infect 2 others with TB before they died. In contrast, if you were treated for worms and you got TB, you would infect approximately 15 others before you died, right? So this is a massive increase in the number of new infections that a buffalo can create over the course of its TB infection if it's dewormed. And so this sort of unanticipated outcome of treatment arises simply because the treatment extends the lifespan of infected individuals. So what's a really important individual level benefit, which is living longer with your TB infection and controlling it, leads to a population level consequence. And so this complicates our idea that we can use simple deworming as a tool to sort of manage TB both at the individual and population scale. And so this is sort of what blending sort of ecological ideas with biomedical theories, which is the source of sort of our ideas about immunological suppression of these worms, can tell us and sort of guide us towards more sustainable solutions potentially for using drug treatment as a control measure. So it's not as simple as those win-win scenarios that people have described. So the second part of this story is about natural host resistance. This part of our work really arose from observations we made over the course of this longitudinal anthelmintic treatment study. So what we found was that these buffalo, much like that sheep graft that I told you about, have this natural resistance to worm infection. So some individuals control their worms naturally and some don't. And that's illustrated here is when we initially catch these buffalo and we sort of quantify the number of eggs they're shedding in their feces as a proxy for how many worms they have, we see this classical aggregated distribution where most individuals don't have that many worms, but some individuals have a lot of worms. This is the worm eggs that we quantify. And when we looked at this in more detail. What was surprising is that we found that there was highly consistent as to whether an individual was shedding a lot of worms or a few worms over the whole 4-year course of the study. So this was highly repeatable. You were either a high shedder or a low shedder. And that was potentially a very natural trait. And so if we just think about these animals in 2 classes as low shedders, 60% of the population was in this low group, and 40% of them was in this high group, where we use this cutoff of about 100 eggs per gram shedding as sort of the line dividing this, which in cattle is an indicator of sort of more clinical versus less clinical symptoms of worm infections. And when we probed these 2 categories in more detail, we found that the low shedders versus high shedders was actually linked to the immune response of worms that they mount in their gastrointestinal tract. And so, for example, we looked at eosinophils and mast cells, which are key immune cells that are in the gut mucosa that help protect against these worm infections. So essentially, they help defend against invading worms. And we see that low shedders, so individuals who are producing very few worms consistently over time, have significantly more of these cells in their intestine. So this phenotype seems to map on to an underlying sort of immunological difference in the magnitude of their defenses against worms at the site of infection in the intestine. And so also our preliminary data suggests that there's a genetic basis to these phenotypes that we see. So borrowing from a wide range of research that has been done in Soay sheep, where they've linked different gene regions or variation in different gene regions to variation in resistance to worm infections, we focused in on this gene, interferon gamma, which is an immune gene, and particularly this locus here, BL4, and variability at that locus we have seen is related to variation in the amount of worm eggs you shed. So our preliminary evidence suggests that this phenotype that we see of being low and high and having these varying immune defenses indeed has a genetic basis, right? And we think this equates to animals that are resistant to their worms naturally versus animals that are non-resistant. So we simply went back to our longitudinal data then and asked, well, what does this phenotype mean for the outcomes of TB infection throughout our study? And so essentially, now we're dividing the individuals as to whether they're this low phenotype, so relatively resistant to worms, or a high phenotype and relatively non-resistant to worms, and we're accounting for whether they got that treatment. But we're asking the same questions. How does this affect the probability that you'll get TB infected over the course of the study, and then the mortality experienced by individuals after TB infection? So in a nutshell, what we found was that the worm phenotype, or your likely being high or low, resistant, non-resistant, had no impact on your likelihood of getting TB infected. Until recently, we actually had never found something that explained your likelihood of getting TB infected. And one reason might be that it's related to the sort of very massive social structure. These animals live in groups that, like in the thousands. TB is a respiratory disease pathogen that's sort of transmitted nose to nose, so there's a lot of contacts. And so it requires a robust immunity to overcome that, continuous exposure. So worm phenotype was not related to your TB infection risk, but like treatment, we found that your worm phenotype was indeed related to your the severity of the TB infection, so your likelihood of dying of TB. And in this case, again, the x-axis is the time since initial TB infection till death or the end of the study and the proportion dying. So we clearly see here that if you're a low phenotype animal, so relatively resistant, you're much more likely to die. So essentially, you're 5 times more likely to die of your TB infection if you're naturally resistant to worms. And at first glance, this was quite surprising to me because if we treat you for your worms, you're less likely to die. If you're naturally resistant to the worms, you're more likely to die. So the question is why? Why does having low worm infections lead to these 2 different outcomes? So clearly the mechanism of having low worms matters. And so the other really interesting thing here is that these resistance effects, so that the low-high outcome, was present irrespective of whether the worms were actually there. So this graph is a little bit busy, but again, this is time in days since TB infection, the proportion surviving, and now I've just plotted the treated animals in red, the untreateds in blue, but also divided them into whether they're the high phenotype, so non-resistant, or the low phenotype. So to orient you, one thing we see very clearly is that if you're a control animal that's not treated and you're the low phenotype, you're the worst off, right? So cumulatively, 44% of them died of their TB infection over the course of the study. But the interesting thing we see here that if you're treated, so you didn't have active worm infections, there's still a difference between high and low individuals. With low individuals, 14% of them dying over the course of the study versus none of the treated high individuals dying. So this suggests that even if you're not actively co-infected with worms, this resistance trait is sufficient to increase your mortality to TB. So it's sort of like, it's not the worms, it's the worms and the ghosts of the worms, right? So potentially this evolutionary response over time to controlling your worm infections leads to paying a price when you're infected with tuberculosis. So more recently, we've been interested in why is this the case, right? So why does natural resistance actually increase mortality of TB-infected individuals while treatment reduces mortality? Why is this different? And so to start getting at that, at the end of our study, we partnered with Kruger National Park, who does every 10 years a TB survey that's lethal, just to understand what is the prevalence of TB and how is TB changing throughout the park to enhance their management strategies. Luckily, the end of our study coincided with their 10-year survey. So they used a subset of our animals as part of their survey because we had a history of TB infection understanding of these animals. So we were able to use these animals to look at the pathology and progression of TB in vivo and ask how do these traits, so being treated or not, or being naturally resistant or not, how do these affect the pathology of TB when we look at the tissues. And so just to sort of orient you a little bit to what we would be looking for, so this is an example of human TB, so Mycobacterium tuberculosis, but bovine tuberculosis sort of acts in the same way. So this is primarily a respiratory pathogen. So you inhale TB bacteria, and they move to the lungs, which is the primary site of infection, and that's where they replicate. And that's where the host mounts this primary immune response. And the immune response to these worms, it comes in the form of this granuloma, so essentially a lesion, which is this combination of immune cells that encapsulate the bacteria, right? And they basically stop it from replicating. And so that's why you get a chest x-ray when you might have tuberculosis, right? Because these lesions or granulomas can be quantified, right? But when the host has poor immunological control, these lesions can burst, right? And then the bacteria are released and they can replicate and then you form new lesions. And ultimately, the bacteria can disseminate to other parts of the body, including the lymph nodes and throughout the animal. And ultimately, this disseminated TB is what leads to mortality. So it all starts with poor immune control in the primary site of infection. So by looking at the lungs and the lymph nodes of our study animals, we can actually look at and assess the degree of immune control they had by asking things like how many lesions do they have in their lungs, what are the immune cells in those lesions, and what stage of the disease do they appear to be in. So we asked some of those questions by looking at lung and lymph node tissue in our euthanized animals of different types. And so I'll just show some example slides here of these results. So when we look at the lungs, which are the primary site of infection, we see that individuals that are low shedders, so they're naturally resistant to their worm infections, have significantly more lesions in their lung. So this indicates poor control of your TB infection in the primary site of infection. These low individuals also are 7 times more likely to have advanced disease controlling for the timing of initial infection. So this suggests that these low or naturally worm-resistant animals are just poor at controlling TB in their lungs. In contrast, the treatment has little to no effect on TB control in the lung, right? So even though treatment seems to lead to this improvement, it's not because there's variation in disease control in the primary site of infection. And so then when we look at the lymph nodes, that tells us more about disseminated infection, so the likelihood that TB is getting out to other parts of the body. Here we see a huge impact of treatment. So control animals have significantly more positive lymph nodes. If you just look at different lymph nodes across the respiratory tract, et cetera, more of these lymph nodes are turning up with TB infection. They're also 12 times more likely to have advanced stage disseminated disease. So the treatment is controlling dissemination of your TB infection. And interestingly, the worm phenotype, whether you're high or low, seems to have very little impact in the lymph nodes. So what's sort of clear to us is that the treatment effects seem to manifest in the lymph nodes, and that relates to dissemination, where the resistance effects manifest in the lung, and it relates to something that's about the primary site of infection. So this is sort of our first piece of evidence that these mechanisms are distinct, and it leads us to hypotheses about particularly what's going on in terms of resistance and how resistance is driving this differential response to TB. So a current hypothesis that we're investigating is that it's a constitutive trade-off at the cellular level that might explain these resistance effects. And to sort of explain this, I'll introduce you to the C57 black 6 mouse and the BALB-C mouse, right? So these 2 mice that are commonly used in biomedical studies are well known to be worm susceptible on the one hand and worm resistant on the other. And the difference, one immunological difference between these 2 mouse strains is that worm susceptible mice tend to have immune cells called macrophages that are biased toward M1 type. So this is just a type of macrophages. Macrophages are important because they're the key immune cell that are in that granuloma I talked about, right? So they're very, very important in TB control. Worm susceptibles have M1 bias. Worm resistance have this M2 bias, which is a slightly different type of macrophage. Well, recent work in mouse models actually show that for M. tuberculosis, at least, that M1 macrophages play a central role in clearing mycobacteria from the lungs, and they're really good at it, but M2 macrophages are not good at it, right? So these are the same, basically the same cells, but a different sort of subtype. And now we know that one of these subtypes is better, the other one is compromised in their ability to do this, and that might explain why worm-resistant BALB-6 mice are more susceptible to tuberculosis. And it might also help us explain what's happening in these buffalo for Mycobacterium bovis. So we're currently exploring this hypothesis in the buffalo system. And so just to kind of summarize what we know, so we know that both having worms and resisting them have consequences for the host response to microbial infection. I think before our work in buffalo, there had been sufficient biomedical studies in rodent models showing that if you have worms, it has an impact on TB. But now we see that it's essentially the same thing, just resisting worms, even in the absence of worm infections. And I think that's a really important result because it says that sort of past coevolutionary history between animals and one set of parasites can have an impact on sort of their interaction with other parasites in the future. Like in our system where TB is an invasive pathogen that these buffalo never experienced, well, now being resistant to worms, which they co-evolved with, is actually leads to negative consequences in the presence of TB infection. So our ongoing work in this system is trying to understand the mechanisms that underlie this natural resistance effect, starting with this hypothesis that there might be an M1 versus M2 macrophage bias in these buffalo that generate this effect, like what has been suggested in mice. And we're also interested in understanding the natural resistance effect and whether it allows us to identify individuals that are high risk for severe TB disease, right? So could this be a general marker of severe TB disease beyond buffalo? So we know that there's variation in natural resistance to worms in human populations. It definitely exists in domesticated animal populations. And maybe this sort of your resistance to a totally different parasite can actually be predictive of your risk of severe disease for another pathogen. And so we're interested in how general this is. And finally, we're interested in how the interaction between worm resistance and TB actually influence host and pathogen evolution, right? So in our system, TB has invaded a system where the hosts have co-evolved with worms. So actually 60% of the population is relatively resistant to worm infections. So can TB actually drive selection towards more, more non-resistance in this population? So what's the evolutionary interactions that might be happening between worms and tuberculosis? Because it's not strictly an ecological interaction, which is how we started this work. And then it might fuel pathogen evolution as well, potentially. So in the last less than 5 minutes I have, I wanted to talk a little bit about sort of my path to this worms, germs, and buffalo topic, and maybe sort of studying the ecology of infectious diseases more generally. And so I like to say that it started from this being a pre-med to WASP, social WASP, to what I do now. But it really, maybe when I thought about this, how to sort of present this, I thought one place it started maybe was with my high school, which was a quite unusual high school. It was called the High School for Health Professions. If you can imagine, it was always a mouthful to tell people that. But I found this quote by the High School for Health Professions, was a high school in Houston, Texas, which is where I grew up, that was the brainchild of Michael E. DeBakey, who was a heart surgeon in Houston. And this was a quote from his son, where he said, what Michael E. DeBakey really wanted to do was to have a school that looked like Houston looked, right? That reflected the diversity of Houston, and a huge impact that this school had, right? It was sort of training students or making them aware of the medical professions, but it was a school where science was important, right? And you were taught that you could excel and enjoy science. And so that's what, with a group of, right, so a large diversity of students, right? So it was an exciting place to interact with others and to learn about science at that young age, right? And to learn that you could excel at it. And so it's really the unique vision, I think, of individual people like Michael E. DeBakey, right, who said, I have this idea to make a school where this is what we do, and we want to bring all types of people to this school. That surely had an impact in how I viewed science. So from there, I left high school, right, really interested in science and thinking, well, I'm going to be a pre-med. And I was, in fact, a pre-med when I got to undergrad. And being enthusiastic about science, I think, led me in the first few weeks of class as a freshman to apply for all these jobs in labs, right? To be like, okay, I'm going to get some experience working in biology labs to really understand what I want to do and write and prepare for medical school. So I applied for all of these jobs, and I got none of them, right? So this is including a job where I was going to be a dishwasher for a Drosophila lab. And I was thinking, I didn't even get that job. Like, what's going on? And so one day at lunch, where essentially in the sort of cafeterias, faculty often sat at tables for lunch. And one of the faculty advisors in our residential college was Joan Strassman, who's a biologist who studies evolution and behavior. And it was the day that I didn't get this job that I was just saying at lunch, I applied for all these jobs and I didn't get one. And she said, why don't you just work in my lab? Right? So here it took one individual, right, to give me this opportunity when I was just like, oh, I don't know anything. I didn't get all of these jobs, including a dishwasher. And she invited me, right, without any information, to come work in her lab. And I ended up working in that lab for 4 years as an undergrad, learning about social evolution. And so this really sort of changed my perspective about science. Although I went to a high school that emphasized science, I had no idea what ecology and evolutionary biology was till I was in this lab. And I thought, wow, right, this is so exciting. And it changed sort of my viewpoint and my trajectory about how I thought about biology. And also how I thought about how people who liked biology actually implemented that. Like, to me, if you liked biology, you could be like a doctor, right? And then I thought, no, I could do research and that would be my life. So this had a huge influence. And so for those 4 years, even though I worked on, this was my senior thesis, I worked on things like, social behavior in paper wasps. It still sort of, I think, instilled in me this interest and enthusiasm for research, right? For generating new knowledge, thinking up ideas and being able to test them, things I didn't know that people got to do for their careers. So this had a huge impact in changing my trajectory from going to medical school to applying for ecology and evolutionary biology programs. And I have never regretted that decision. So early in graduate school, I figured out by reading, your first year of graduate school, you're reading all these papers. And I read this paper by Richard Alexander on the evolution of social behavior because that's what I went to grad school interested in. And I realized after reading this paper that there was a link between social behavior and infectious diseases. And that was it hooked me, right? And I thought, oh, well, my interests are not that different. I don't have to give up the interest that I did have in sort of pathogens and things like that, and the interest that I had in social behavior. And unknown to me at the time, Dick Alexander was actually the supervisor for my undergrad mentor. So there was this weird connection that I didn't know. But again, I think this was sort of this seminal trajectory that I would like to say basically started with the opportunities or big ideas that, and small ideas that individuals had. So I guess I just wanted to end in saying that sort of as a faculty member now, I look back and I think that people's, just one person can make a difference, I think, in changing the face of science. So DeBakey's idea to have this school, which I think from when he initially had the idea to when the school was founded was 10 to 15 years. that's changed the lives of countless scientists. Joan Strassman giving me a chance in that lab has changed the face of my academic career. So it really just takes one person. And so as I've had the opportunity at UGA to try to contribute to that, right? So to change the face of science one at a time for individuals, right? By not only sort of integrating human diversity into what we do, by being sure that we're trying to reach out and recruit students from all walks of life into our departments and into our programs, but also combining that with the fact that, you know, we also want disciplinary diversity. So particularly in the area of infectious diseases, which I think is one of the grand societal challenges that we face, that integrating human diversity and disciplinary diversity, so whether it's biomedicine or public health or ecology and evolution, bringing those ideas together with a diverse scientific population will really help us tackle at least the challenge of infectious diseases. And we at UGA in our programs are trying to do that sort of one person at a time. And last, I would just like to thank you guys for your attention and thank all my collaborators on the Buffalo Project and many students and staff who have really contributed to this work. Thanks. Yes.
Well, thank you very much for your talk. It was really super interesting. I'm glad that we rescued from medical school.
Yes, I'm glad about that every day.
I was going to ask if you have any follow-up of the offsprings of your buffalo population. If you have any data will happen, you know, later on, the next generation of those.
Oh, you mean their offspring?
Yeah.
So unfortunately, we couldn't track their offspring simultaneously. So during the course of the experiment, the adults had the radio collar, and they were giving birth, but we didn't have collars or the ability to track their offspring. So we don't know what's happening in the next generation. But our work now is trying to potentially use wide-scale cross-sectional sampling to understand things like the heritability of this trait, right? Without tracking, you know, parents and offspring, which is a little challenging in our system. Yeah. Oh, Kate.
I was wondering if you, for your fecal egg count phenotype, so it sort of nicely groups into low and high, but I was wondering, have you looked at it continuously, and do you see similar patterns, or is it more of a kind of stark contrast?
Yeah, so continuously, like I've looked at it, but I think we don't have enough data because it's aggregated, first of all. So I think essentially you get a weak signal when it's continuous, and so when you dichotomize it, you pull out the signal. But I'm hoping that in our larger scale sampling, we'll just sample a large enough group of animals that we can start pulling out like a continuous effect, which I think we should be able to do that with a large enough sample size. But in this respect, right, the sample of the low and high and their effects, it's actually the low and high that are TB infected. So at that point, there's not very many.
Why don't we go ahead and thank Dr. Ezenwa one more time. If you have other questions, come on.